Hello, I'm Aditya Iyer, and I will be giving a rundown of my poster titled K-Spheres, a novel methodology for clustering differentiating stem cells. This is coming from the labs of Dr. Jeremy Purvis and Dr. Lidong Lee at the University of North Carolina at Chapel Hill. Let's begin with the motivation and relevance of our method. Understanding the molecular mechanisms by which embryonic stem cells differentiate into germ layers is a key goal in regenerative medicine. A major challenge in this effort is that stem cells simultaneously undergo two interdependent processes with distinct dynamics. 1. Differentiation, which is sequential changes in the molecular states of cells during development. And 2. The cell cycle, a cyclical process by which one cell gives rise to two daughter cells. Given this interdependence, we propose that stem cells differentiate via a series of cyclical molecular states, as shown on the bottom. This is in contrast with the purported acyclic differentiation trajectory model, as shown on the top. Our hypothesized approach can power clustering and trajectory inference methods for identifying germ layer lineages and cell cycle phases. Each cyclical state may correspond to a germ layer lineage or a subtrajectory of a germ layer lineage, such as early endoderm. These states may also correspond to a population of pluripotent cells. Most current trajectory inference topologies, however, don't account for our hypothesis, as they are mostly branch-based. The ones that can produce cycles don't possess interpretable geometric properties to characterize the trajectories. We demonstrate that K-spheres can fill this gap by applying it to a dataset of differentiating stem cells. Before we delve into our methodology, let's briefly look at the experimental procedure used to generate our data. We seeded human embryonic stem cells to differentiate into one of the three germ layers, the endoderm, mesoderm, and ectoderm. We also seeded cells to remain pluripotent as a control. We left our cells to differentiate for discrete time intervals of 24, 48, and 72 hours. It's important to note that although we seeded cells to differentiate into pre-specified lineages, they likely contain impurities due to the interactability of directed differentiation. On each collection of cells, we performed 12 rounds of iterative indirect immunofluorescence imaging, or 4I for short, using a curated set of proteomic biomarkers associated with differentiation and the cell cycle. 4I is a novel multiplexed microscopy technique for obtaining dozens of proteomic features from single cells, increasing the dimensionality of our data. We applied a series of pre-processing steps to the collected data samples, and in total, our final data set consisted of 20 Z-normalized fluorescence intensity measurements for 109,826 cells. We visualized our data by generating 2D FATE embeddings. FATE, spelled P-H-A-T-E, stands for Potential of Heat Diffusion for Affinity-Based Transition Embedding, and it is a relatively new manifold estimation technique that provides an accurate denoised representation of both the local and global structure of a data set in the required dimensionality without imposing any strong assumptions on the structure of the data. We produced overlays of the intended differentiation lineages, as shown on the left, and the length of differentiation, as shown on the right. As you can see, neither overlay contains a lineage or length of time category that encompasses a complete cyclic structure. This contradicts our cyclical differentiation trajectory hypothesis. The absence of cyclic structure supports our assumption that several of our seeded cells did not differentiate as intended. Therefore, we must retrospectively annotate cells to identify the lineage to which each belongs. K-spheres can aid in this effort. Now on to the formulation of K-spheres. The goal of the method is to partition the data into subsets that exhibit spherical patterns. The algorithm to achieve this is similar to Lloyd's algorithm for k-means, as it contains a cluster initialization step, followed by a step that updates the parameters, and a step that reassigns the cluster label based on distance measures. This is repeated until convergence is reached. The key difference from Lloyd's algorithm is that K-spheres considers distances to low-dimensional spheres instead of distances to centroids. The low-dimensional spherical components are computed using spherical principal component analysis, or SPCA for short. This is parameterized by a center, radius, and low-dimensional affine subspace. These SPCA properties help K-spheres capture the geometry that underlies differentiation and the cell cycle in a biologically interpretable manner. For example, shifts in the cluster center and radius can inform us of changes in the biomolecular content and brevity of a cell cycle, respectively. We place K-spheres in a family of cycle-based clustering methods. These include mixtures of Fisher-Gaussian, or mixtures of FG kernels. Mixtures of FG kernels are a probabilistic or soft clustering generalization of K-spheres, similar to how Gaussian mixture models are a probabilistic generalization of K-means. Additional cycle-based clustering methods can be conceived with the potential to outperform K-spheres at capturing the cell cycle-induced heterogeneity of differentiation. For example, spherical principal component analysis can be replaced with a method for ellipsoid fitting, as a cell cycle may not be perfectly spherical in nature. Finally, we move on to our results. We compared K-spheres to K-means in Gaussian mixture model clustering. With each method, we used the SOC17, EOMs, and GATA6 markers to identify the cluster that most likely corresponds to the endoderm lineage. The clusters are overlaid on the fate embeddings. As you can see, only the K-spheres cluster seems to encompass a complete cyclic structure. Therefore, given our cyclic differentiation trajectory hypothesis, only the K-spheres cluster is likely to encompass the near entirety of the endoderm lineage. Once the clusters are obtained, we can map out all the phases of the cell cycle within each one. It is worth noting that under our hypothesis, the endoderm lineage may consist of several sequential cyclical subtrajectories. However, our data appears to only contain a single cyclic structure for the endoderm. Looking ahead, we can apply K-spheres as well as other cycle-based clustering methods to new datasets, both biological and non-biological, that feature cyclic structures. This concludes our presentation, and thank you for listening.